There are enough differences in the Hox gene expression pattern between insect and crustacean. When we were talking about the development of thoracic legs and insects not having leg in abdomen, what I told you was that the presence of distalless gene DLL causes the emergence of thoracic legs, while the UBX and the abdomen A expression prevents or stops the expression of DLL. Therefore, in abdominal region, the DLL gene expression is stopped by the proteins produced by UBX and abdominal A. So this pattern of the expression of Hox gene is different than the expression in crustaceans. The crustaceans have a different Hox gene expression pattern. In crustaceans, among them also, there is a great difference in the expression of Hox gene pattern. In most families of phylum Arthropoda, UBX protein generally does not inhibit the distalless gene. However, in the insect lineage, a mutation occurred in the UBX gene wherein the original 3' end of protein coding region was replaced by a group of nucleotide encoding a stretch of about 10 alanine residue. So it is because of this mutation that happened in the UBX gene in class Insecta that the different Hox gene expression pattern evolved and UBX inhibited the expression of distalless gene. Also, this was what we talked about in invertebrates. We took the example of phylum Arthropoda members. Now, change in Hox gene expression between body segment is also observed in vertebrates. Hox gene in vertebrates determine the type of vertebral structure formed. The thoracic vertebrae, for example, have ribs, while the cervical, that is the neck vertebrae, and the lumbar vertebrae do not have ribs. So, it is the Hox gene expression in the somite that caused the different type of vertebrae to be produced. Similarly, another example is that of the snake and why the snakes don't have any legs. The snakes have evolved from the lizard. We know that Hox gene is responsible in vertebrates for providing them the limb identity. They are important in providing the positional identity. This we have done in tetrapod limb development, how Hox genes were responsible, and we know that Hox gene in humans are clustered in 4 chromosomes, and they have been designated as Hox A, Hox B, Hox C, and Hox D. We have learned in earlier class that how in tetrapod limb development the Hox genes are important for giving the limb identity, whether the limb will be forelimb or hind limb, and also they were important along with TBX in determining the position from where the limbs will originate. So snakes, we know, have evolved from reptiles, from lizards. Both are reptiles, and snakes have evolved from lizard. We know lizards have both forelimbs and hind limbs. So, how the snakes have lost their legs? The loss of leg has occurred slowly and gradually in two-step process. Both the paleontological and embryological evidence support the view that the snakes have lost their legs in two-step process. They have first lost their forelimb and then they lost their hind limbs. The fossil snakes with hind limbs have been discovered, but no fossils where forelimb present has been found. Most of these snakes, such as vipers, they are totally without limbs, while some primitive snakes like boa and python, they have pelvic girdles to which the legs are attached. So in them, in python and boa, the pelvic girdles were present, and these girdles had the rudimentary femur. Femur is the long bone of hind limb. The missing forelimbs in snake has been explained by the Hox expression pattern in the anterior portion of the snake. In most of the vertebrates, in most of the vertebrates, the forelimbs are formed anterior to the most anterior expression domain of Hox C6. Caudal to that point, Hox C6 in combination with Hox C8 help to specify the vertebrae to be thoracic. During early python development, Hox C6 is not expressed in the absence of Hox C8. So, the forelimbs do not form. The combination of Hox C6 and Hox C8 is expressed for most of the length of the organism. telling the vertebrae to form the ribs throughout most of the body. The loss of hind limbs has occurred by a very different mechanism. Hind limb buds begin to form in some snakes, such as python, but they do not produce anything more than a femur. This appears to be due to the lack of sonic hedgehog expression by the limb bud mesenchyme. Sonic hedgehog is needed both for the polarity of the limb and for the maintenance of apical ectodermal ridge. Thus, we can say the python's hind limb bud lack the AER, and the phenotype of python hind limb resemble that of a mouse embryo with some mutation of sonic hedgehog. Alteration of Hox gene expression can change one type of vertebra into another. The mutation of a Hox gene can produce atavistic condition. Now, what is atavism? Atavism means the inheritance of some ancestral characters. So, mutation in Hox gene can produce atavistic condition wherein the organism resembles an earlier evolutionary stage. Major evolutionary changes can be correlated with alteration of Hox gene expression in different part of the embryo. Like occasionally in nature, we see the animals that are born with mutation can cause them to exhibit, to show ancestral trait. For example, we can see that snakes where some mutation in Hox gene has occurred, we see that they are born with legs. Another example of atavism in nature is we can see whale with tiny hind limbs or horses with toe. It means that some mutation or alteration has occurred in their Hox gene clusters, and because of those mutation, some ancestral character or trait or phenotype has been exhibited in the animal. Another example is where atavistic condition is seen are the humans with tail. These traits, which occur in both plants and animals, are called atavism or evolutionary throwback. Some animals also normally retain the ancestral trait in a very reduced form, such as the pelvic structure in whale or the sightless eye of the blind mole rat. In them, the ancestral character is kept preserved or conserved in their DNA but in reduced form. and these later features are called the vestigial structures. Both atavism and vestigial structure tells us about the evolutionary history of an organism. In normal embryonic growth, the genes that control development are switched on in a set pattern so that different body parts of or the body system develop in sequence. Developmental genes effectively instruct the embryonic cells what body part to grow, where to grow, as well as when to start and when to stop the growth process. The genes that control the basic development of all animals are surprisingly similar. As we had talked in previous lecture about the unity of type. So I was telling you the basic development of all animals are similar, having been conserved over millions of years. For example, the PARK6 gene control the development of eyes across the animal kingdom from fruit fly to human. This we have discussed in previous lectures. So, to sum up, we say that the forelimb forms just anterior to the most anterior expression domain of Hox C6. Hox C6 plus Hox C8 causes the formation of thoracic vertebrae from the ribs. During early python development, Hox C6 is not expressed in the absence of Hox C8, so the forelimbs do not form. Rather, the combination of Hox C6 and Hox C8 is expressed for most of the length of the organism, telling the vertebrae to form the ribs throughout most of the body. The hind limbs but do begin to form in python, but they do not make anything more than a femur. They do not elongate into whole hind limb. Why? Because of the lack of a morphogen called sonic hedgehog, which is important for the development of limb bud. Sonic hedgehog is needed for both the polarity of the limb as well as for the maintenance of apical ectodermal ridge. This we have done in tetrapod limb development, that how from the limb bud various morphogens are released and the limb develops in anterior-posterior dimension, in dorso-ventral dimension, in proximal distal dimension. This we have done. So just revise that. Will not go into detail about the sonic hedgehog and apical ectodermal ridge. So to sum up, we can say that sonic hedgehog is needed for both the polarity of the limb as well as for the maintenance of AER, and the python hind limb bud lacks the apical ectodermal ridge. When we were talking about the alteration or the mechanism by which the Hox gene expression is altered, we have discussed that before we had discussed that the number of Hox gene, if it is one or more, then also it causes the change in or it causes the descent with modification. So, or we can say that it also causes the deviation from unity of type. If in simple organism one copy of Hox gene is present, it will exhibit one type of expression pattern. If the same Hox gene is present in multiple copies in other organism, then the expression pattern will be different. So change in Hox gene number also causes the alteration in an individual's structural plan. The number of Hox genes play an important role in permitting the evolution of complex structure. All invertebrates have a single Hox complex per haploid genome. For example, in Drosophila we have seen that chromosome number 3 has got 8 Hox genes, like 8 homeotic genes. The homeotic genes are present in two clusters. The first cluster is called the Antennapedia complex, and the second cluster or complex is called the Bithorax complex. Antennapedia complex has the labial, uh, deformed, sex comb reduced Antennapedia, like that, 5 genes, Hox genes, and the thoracic complex, bithoracic complex has 3 Hox genes, namely ultra bithorax, abdominal A and abdominal B. So all invertebrates, they have single Hox complex per haploid genome. In simplest invertebrates such as sponge, there appear to be 1 or 2 Hox gene. More complex invertebrates such as insect have numerous Hox gene in this complex. Comparing the Hox gene of chordate, arthropod and mollusk, it suggests that there was a common set of 7 Hox gene in our bilaterian ancestor of the protostomes and deuterostomes, and in invertebrate deuterostome like echinoderms amphioxus, there is only one Hox complex, which looks very much like that of the insect. But as the time passed, the earliest vertebrate evolved and there were 4 Hox complexes. The transition from amphioxus to early fish is believed to be one of the major leap in the complexity during evolution. And this transition involved the evolution of head, the neural crest, new cell types such as osteoblast, that is those cells which form the bone, odontoblast, the brain, and the spinal cord. How does the new cell type formed? It involved the duplication and divergence of the genes. For example, the DLL gene originally has one copy in amphioxus but has about 5 to 6 closely related copies in vertebrates. The DLL homologs have found new function in modern vertebrates, that is, they expressed in mesoderm and they expressed in the forebrain. Although it remains unproven. Why? Because we don't have any fossil record, fossil evidence of the bilaterian ancestor. So though it remains unproven, it is possible that new type of Distillers gene could have caused the migratory ectodermal cell of Amphioxus to evolve into neural crest cell.